Welcome back to Concepts of Biology. We'll read section 4.4, Fermentation. By the end of this video, you will be able to discuss the fundamental differences between anaerobic cellular respiration and fermentation, and describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. In aerobic respiration, the final electron acceptor is an oxygen molecule, O2. If aerobic respiration occurs, then ATP will be produced using the energy of the high-energy electrons carried by NADH or FADH2 to the electron transport chain. If aerobic respiration does not occur, NADH must be reoxidized to NAD positive for reuse as an electron carrier for glycolysis to continue. How is this done? Some living systems use an organic molecule as the final electron acceptor. Processes that use an organic molecule to regenerate NAD positive from NADH are collectively referred to as fermentation. In contrast, some living systems use an inorganic molecule other than oxygen as a final electron acceptor to regenerate NAD positive. Both methods are anaerobic, meaning that they do not require oxygen to achieve NAD positive regeneration and enable organisms to convert energy for their use in the absence of oxygen. Lactic acid fermentation. The fermentation method used by animals and some bacteria, like those in yogurt, is lactic acid fermentation. This occurs routinely in mammalian red blood cells and in skeletal muscle that has insufficient oxygen supply to allow aerobic respiration to continue, that is, in muscles used to the point of fatigue. In muscles, lactic acid produced by fermentation must be removed by the blood circulation and brought to the liver for further metabolism. The chemical reaction of lactic acid fermentation is the following. Pyruvic acid plus NADH gives us lactic acid and NAD positive. The enzyme that catalyzes this reaction is lactate dehydrogenase. The reaction can proceed in either direction, but the left to right reaction is inhibited by acidic conditions. This lactic acid buildup causes muscle stiffness and fatigue. Once the lactic acid has been removed from the muscle and is circulated to the liver, it can be converted back to pyruvic acid and further catabolized for energy. Figure 4.16. Lactic acid fermentation is common in muscles that have become exhausted by use. Here's a quick question. Trematol, a metabolic poison found in white snakeroot plant, prevents the metabolism of lactate. When cows eat this plant, trematol is concentrated in the milk. Humans who consume the milk become ill. Symptoms of this disease, which include vomiting, abdominal pain, and tremors, become worse after exercise. Why do you think this is the case? Share your answers in the comment section below. Alcohol fermentation. Another familiar fermentation process is alcohol fermentation, which produces ethanol, an alcohol. The alcohol fermentation reaction is the following. Pyruvic acid leads to carbon dioxide and acetaldehyde with the introduction of NADH shifting to NAD positive leads to ethanol. Figure 4.17. The reaction resulting in alcohol fermentation is shown. Hi, if you found this video informative, give it a thumbs up, subscribe, and share the link. We want these free textbook resources to be available to other students. All right, let's get back to reading. In the first reaction, a carboxyl group is removed from pyruvic acid, releasing carbon dioxide as a gas. the loss of carbon dioxide reduces the molecule by one carbon atom, making it acetaldehyde. The second reaction removes an electron from NADH, forming NAD positive and producing ethanol from the acetaldehyde, which accepts the electron. The fermentation of pyruvic acid by yeast produces the ethanol found in alcoholic beverages. If the carbon dioxide produced by the reaction is not vented from the fermentation chamber, for example in beer and sparkling wines, it remains dissolved in the medium until the pressure is released. Ethanol above 12% is toxic to yeast, so natural levels of alcohol in wine occur at a maximum of 12%. Figure 4.18. Fermentation of grape juice to make wine produces carbon dioxide as a byproduct. Fermentation tanks have valves so that pressure inside the tanks can be released. Anaerobic cellular respiration. Certain prokaryotes, including some species of bacteria and archaea, use anaerobic respiration. For example, the group of archaea called methanogens reduce carbon dioxide to methane to oxidize NADH. These microorganisms are found in soil and in the digestive tracts of ruminants such as cows and sheep. Similarly, sulfate-reducing bacteria and archaea, most of which are anaerobic, reduce sulfate to hydrogen sulfide to regenerate NAD positive from NADH. Figure 4.19. The green color seen in these coastal waters is from an eruption of hydrogen sulfide. Anaerobic, sulfate-reducing bacteria release hydrogen sulfide gas as they decompose algae in the water. Other fermentation methods occur in bacteria. Many prokaryotes are facultatively anaerobic. This means that they can switch between aerobic respiration and fermentation depending on the availability of oxygen. Certain prokaryotes, like Clostridia bacteria, are obligate anaerobes. Obligate anaerobes live and grow in the absence of molecular oxygen. Oxygen is a poison to these microorganisms and kills them upon exposure. It should be noted that all forms of fermentation except lactic acid fermentation produce gas. The production of particular types of gas is used as an indicator of the fermentation of specific carbohydrates, which plays a role in the laboratory identification of the bacterium. The various methods of fermentation are used by different organisms to ensure an adequate supply of NAD positive for the sixth step in glycolysis. Without these pathways, that step would not occur, and no ATP would be harvested from the breakdown of glucose. This ends Chapter 4, Section 4.4, Fermentation. In the next video, we'll read Section 4.5, Connections to Other Metabolic Pathways.